I am Dr.S. Anbuselvi. I am going to deliver a lecture on the mycorrhiza biofertilizers. This is the one type of the biofertilizers based on the fungus. Okay, the fungus also it is involved in the development of the biofertilizers. Okay, so what is the called as the biofertilizers? The living microorganisms. It is used as the fertilizer for a growth of a plant is called as the biofertilizer. Okay, and the next thing is what is called as mycorrhiza biofertilizer. Mycorrhiza biofertilizer is also known as the VAM. VAM means myco means the fungal, rhizo means the root. It occurred to the plants. It is called as the rhizoids. It is leading to the development of a hyphae. So hyphae, it has a boost the development and the spreading of the white root in the soil, leading to the significant increase in the rhizosphere. And these hyphae further penetrate and form arbucles. Okay, arbucles within the root corticles. The VAM fungi form a special symbiotic relationship with the roots and or roots of a plant that had enhanced the plant growth and the survivability of the colonized plants. This mycorrhiza is very useful in organic farming as well as of a normal commercial farming. You can use as a biofertilizer. Okay, what is it called as a mycorrhizae? Myco means fungi, rhizo means root. So within the root, this fungi is present. How it can be penetrate? It is a penetrate into the plants via the soil. Okay, so via the soil means it has entered into the roots of a plant. So VAM fungi, it get the nutrient from the roots of a plant. The plant also get the beneficial from the fungi. Therefore, both of them are called as a mutually benefited. It is called as symbiotic relationship. Okay, and then next thing is what is it called as a VAM? VAM means vesicular arbuscular mycorrhizae. It is called as VAM. The term mycorrhizae was taken from the Greek language. Mycorrhizae means derived from the Greek language. It is the meaning of this. It is called as the fungus root. The term was coined by the Frank in the year 1885. The mycorrhizae is the mutual understanding. That was mutualistic association between the fungi, especially of mycelia and the plant roots. And then VAM, it is called as endotropic. Endotropic means it is living inside the root of the plant. So mycorrhiza, it is formed by the accepted mycocyptus fungi. The VAM, it is very helpful in the nutrient transfer, mainly phosphorus, sulfur, and the zinc. Okay, and then what is it VAM? VAM means it is called as vesicular arbuscular mycorrhizae. Okay, so these mycorrhizae, what more it is there? It is intracellular, obligate, fungal, endosymbiote, and glomerulus. It transfers the nutrients. So what are the nutrients means? It has a phosphorus, zinc, sulfur from the soil to the roots. Okay, and then history of mycorrhiza, if you are taken, the fossil records show the mycorrhizae have been around. It is the longest history is there. It is around 460 million years old to 460 million year old. Fossils also they are taken this type of mycorrhizae. So especially in the underground parts of the soil. So underground parts of the soil have the mycorrhizae fungi. So these fungi very helpful in the growth of the plants. How it can be grow that? So the Robert Hadrich in the year 1840, the first isolated the mycorrhizae find the roots of a pine tree, the needle-shaped pines, you get it that, but did not recognize them as a separate being. And the next thing scientists, yes, Reitzek in the year 1847 described and recognized as the fungal cells associated with the archates. Okay, because it is a grow that, therefore it is associated with the archates. And then the last, finally, the scientist Frank in the year 1885 developed a fungus. Okay, the rooted structure, the fungus rooted structure, it shows the increased growth of the plant when the fungus it is present in mycorrhizae. Mycorrhizae means the fungus it is present or located inside the root system of a plant. Then it is called as mycorrhizae. And the next case, what is the function of this mycorrhizae? This mycorrhizae it shows the symbiotic relationship between the fungi and the plant. So ECM are not obligate mutastasis, that is called as mutually understanding, mutualism, but BAM or the fungi, it impart the benefits to the plant by increasing what ways means. The first point is the nutrient absorption. So what are the nutrient it has absorbed? That is the phosphorus, water uptake, and the tolerance. Tolerance means stress tolerance or the drought tolerance. So these two cases it can support it. The environmental conditions, including even the polluted water or soil, it is not fit for that the plant can grow. Because of this, these important feature in this tolerance activity. Therefore, it is act as a shield. Shield means protect. Okay, so protect against acidity, elemental toxicity, and pathogens. Okay, and then so pathogens means antigens, that a foreign substance like a pathogen or insects, it is affected it is inhibit. Okay, so it inhibit the activity, it act like a protective shield and the defense mechanism against the various pathogens around the plants, especially in the soil. And what are the factors it affect the mycorrhizae formation means? The first important factor is that is of the amount of above the ground plant cover is because the mycorrhizae is present inside the soil. So these factors it is play a significant in the role. But the soil moisture, soil depth, grazing, and pesticides and soil fertility, these are the very minor factors can affect the mycorrhizal growth. And then what are the benefits of this mycorrhizae means? The first point is increased uptake of the nutrients. So nutrient, especially of that is of a phosphorus. And then they have to support that is of a growth of a plant, increase the water surface area, soil surface area. So water holding capacity is more and absorption of nutrient, the less intake of water. So it can support to the growth of a plant. And then the nutrients such as phosphorus, zinc, carbon, nitrogen, copper, and sulfur have to shown to be absorbed and translocated to the host by the mycorrhizae fungi. And the next point is what ways it has a support. It has increased the rootlet size and longevity. So the mycorrhizae plants have longer roots when compared to the ordinary plant. Whenever you are showing that biofertilizer, so what are the differences it shows means? It shows the longer root. Okay, so longer root, therefore more shelter activity also, shielding activity, and than that of a mycorrhizae plant, regardless of whether the mycorrhizae fungi is present or not, automatically it shows the benefit of the mycorrhizae. And then water tolerance. So water tolerance, if you are having this hyphae, they explore the soil for the water and increase the surface area for the absorption of the nutrient. And the same time, the mycorrhizae, they can alter the plant physiology, increasing the stomata resistance. Stomata means that transpiration activity, water absorption. So that also automatically it is increased. Resistance means what happened that the water absorption is very less amount. So it is resulting in what? The water loss. The water loss in plant is, it is the less amount. It is minimized. Okay. So that is called as water loss. And then next thing is tolerance. So tolerance means the fungi are very tolerant to acidity. Acidity means the soil pH. The pH of the soil is around 5.5 to 7. It is suitable for a growth of a plant, but either it is acidic, less than 7, around 3 to 4, or 9 to 10, alkaline pH, that cases it has also affect the growth of a plant. But if a fungi is present in the root, automatically it is tolerant to acidity and elemental toxicity. The polluted water also the plant can grow, and high temperature, the soil, the temperature, that things is especially the plant can grow well nicely, and then the sheath, the root from these condition means from the drought or from the blood, it is protected it. And the lower levels of heavy metals are generally found in this mycorrhizae plant than that is of a non-mycorrhizae plants. And then next point is increasing the seedling survival. These mycorrhizae promote the plant survival rate is more. How? Because of the new seedlings are coming out in that. You are having a put it in the stock, and then you can check it also what happened. The new seedlings can grow very well. And the survival of this inoculated plants can up to how long time it has survived 5 times more than that of the uninoculated plant. Therefore, we are telling that it is have the more shield life. Okay, and then improve the survival is a no doubt due to the combination of this mycorrhizae benefits, including the faster growth to help develop a weed control, overlap the weed control, protection from the pathogens, that is of a plant disease causing the pathogens, it is prevent entry, that is called a protection. The plant defense mechanism is very good, and improve the drought resistance. If the water is not there, the plant can survive. So these cases they can benefit. So these are the some examples of this mycorrhizae biofertilizer. So seed care kit, it is available commercially in the market also. And then what is the relationship between this mycorrhizae and the pathogen? The mycorrhizae fungi have long been known to that defend the root against the soil-borne diseases, pathogens. So what is the relationship between the pathogens and the mycorrhizae means? The main thing is the protective shield. So protective shield, how they have to be means, it is to promote the beneficial activity, the directly response, it produce some compounds to protect against the root pathogens. And this mycorrhizae fungi may reduce the incidence and the severity of the root diseases. The mechanism proposed to explain the protective effective include the development of the mechanical barrier, especially the model of EM to the infection by the pathogens. At the second point, the protection of antibiotic compounds. We are telling that any pathogen enter immediately the antibiotic compounds like a compound, it inhibits the activity of the entry of the pathogens. Here also the protection of antibiotic compound that suppress the pathogen growth. And then next point is the competition for the nutrients with the pathogen, including the production of the cytospores. And then the last point is induction of generalized the coast defense mechanism. Coast means the plant itself, the defense mechanisms against the pathogens. So this is the relationship. But what is the disadvantages in this mycorrhizae means when the nutrient levels in the soil are so low and the fungus cannot extract extra nutrient or benefits, okay, as accelerate to the plant. So in turn for that, the carbon transfer to the fungus, it is called as parasitism. So parasitism means if it is more fungal growth is may occur due to that is of a nutrient, it is inhibit the plant growth. So everything too much is it causes the damage, right? So same thing here, it is also what happened if your nutrient levels are very low in the soil and the fungus cannot extract it. So the nutrient content is present, then only the fungal growth is also more. And then sometimes the plant growth suppression have been attributed to the mycorrhizal colonization. So the plant growth also what happened, it suppress the growth of the colonies. But usually this occur only in the unusual circumstances that affect the plant photosynthesis. So the plant photosynthesis cannot occur, the plant cannot grow, especially of the low light and the cold temperature or the nutrient, especially of the phosphorus. If the phosphorus content is high, automatically it is inhibit the growth of this mycorrhizae. So what is the management we are doing that? The most of the mycorrhizae fungi are carried out in the top, that is a 15 centimeter of the soil. Don't bury it inside more amount on that soil. Okay, so also only that is you are loaded on the topsoil. So preserve the topsoil and microbial activity. It is a fungal, right? So spread on the surface of the soil and do not use any chemicals. If you are using the chemicals, it kills the fungi. And another one more thing is do not over fertilize. Don't put more amount. If you are putting more amount, the bacteria, the fungal cannot grow. And then keep always the native plants, the smaller grains of millets like that native plants, you can grow it with the dust of a mycorrhizae. So what are the benefits of this mycorrhizae? It can produce more vigorous and healthy plants. And then another important thing is it can increase the plant establishment, because already I thought the plant grow well and survival rate is more, and the seedling or especially in the transplanting, especially suppose you are taking the 10 seeds, if you are taken around the 5 to 6 seeds are grown in the normal condition, but this case if you are putting this VAM act as a biofertilizer, around 8 to 9 seed are enter into the seedling stage. Or therefore it is a suitable for seedling or transplanting. And then especially it enhances the flowering and the fruiting. So the yield of a plant, it's more in that is of using this VAM, and then increase the yield and crop quality also increase. And the last point, the improvement in that is of a drought tolerance and allowing that is of a watering reduction. So the water usage is less. Okay, so this is the main benefit in that. And the next case, and optimize the fertilizers use especially of this phosphorus and increase the tolerance to the soil salinity and reduce the degrees disease, reduce or decrease the disease, that is a pathogen entry into the plant is reduced or completely it is block the activity, therefore it acts as a barrier or the protective, okay, or the coast, okay, defense mechanism is well developed using this VAM and contribute to maintain the soil quality and nutrient cycling. And the last one, it has a contribute to control the soil erosion also. So if the plant is root is well fixed, automatically what happened that the soil erosion is also to be, it is reduced. So, and then next thing is come to the isolation. How you are going to isolate the VAM spores from the soil for making the biofertilizer? The first one method is called as sieving method. So this sieving method, what are the requirement means? Soil sample, 500 ml beaker, sieves, that is microspores. You are taking the spores of this mycorrhizae and 710 um, 250 um, 75 um, and 45 um, and Bunsen burner. So these are the things. And the procedure: 200 ml of water taken in a 500 ml beaker. Heat the water to 40 to 50 degree. Add 50 gram of soil. Mix it well and make the suspension. Because any other pathogen is not present in the soil, so you are sterilize it. And after the sterilize it, allow to the settlement. So allow what happened there, it allow the particles to settle down and decant the most of the suspension mainly through the 710 micro seed to remove the larger organic matter and roots. And then second method, if you are taken this, the large organic matter and roots. And then next thing is the flotation method. First one is the sieving method, and then second one is the flotation method. And this method you are taking the soil sample, and then nutrient growth is having using the sucrose solution with the different concentration of 20, 40 and 60%, and then the blender it. And then you have to use the fine sieve. All the case the siever is more probably it is used, and centrifuge and centrifuge tube. You have to centrifuge it and collected it. Okay, and this next procedure, you are taking the fresh soil of the sample from the field, mix well, and you have to make the 20 gram of soil and transfer the soil into the blender and blend it at high speed for 1 to 2 minutes. So the spores completely won't happen, the blend mix mixer. So automatically the spores are attached to the soil particles or root may become free and filter the content, because from the root only we are taking the spores, and filter the content through the fine sieve and wash with a strong stream of water and pour 10 ml of 20% sucrose into the centrifuge tube, followed by the same amount of 40% or 60% of sucrose into the bottom of the tube. And then next case, you have to see that is of a transfer. So collected this for 40 or 60% into the bottom of the tube. After you have blended sieving it, add on to the surface of 20% of sucrose layer. Centrifuge the content for 30 minutes at 3000 RPM. You have to remove the deberries. Deberries means unwanted materials. Everything you have to remove it, which accumulate the interface of 20 to 40% and 40 to 60% of sucrose, because it is a density gradient centrifugation like this. Therefore, we are using the sucrose. And gently wash the spores present on the fine sieve with a strong stream of water. So the sucrose should be completely removed, and collect the spores and observe under the microscope. So how you have to observe it? You have to take the fungal, and that fungal you want to multiply more copies you want. You can put it in the potato textose agar media and fungal isolation and screening procedure. From that you have to confirm this vesicular articular worm is present or not. Okay, so this is the procedure. After this inoculum production, you have to go for the mass production. So mass production, if you are taken it, you have to put it in this fermenter, and then you have to culture it. You are getting a more amount of this back. So being this obligate side one, the AM fungi could be the mass produced only by the presence of what? Living roots. So AM fungal association are very universal and have been reported in all terrestrial plants. So these can be reproduced in a wide range of host plants. There are several techniques are reported for the mass production of the AM inoculum. Okay, now we are coming to the in vivo culture. So AM fungi are grown on the roots, especially of a greenhouse plants. If you are taken, chop the mycorrhizae roots and often mixed with the growth media. The growth media, especially for the hyphae and the spores, it is used as a source of inoculum. So source of inoculum is hyphae and the spores. And the soil could be replaced by inert substance such as vermiculite, perlite, sand, or mixture of this for a crude inoculum protection. This is the carried out in the in vivo condition. So here the mass production of worm inside that is of a brick tank, or mass production of worm inside carried out in that is of a cement tank. So this is the feature. So first thing is the tank is get ready for mass multiplication of this worm. So sprinkling of this water in the tank with the vermiculate. First step is sprinkling of this water. First thing, water with the vermiculate you have added, and then mixing or making a that is of a furrows. Furrows means a small rows. Okay, the two that is of a sow the maize seeds. So maize seeds you have to make the furrows, and then next step sowing the seeds in the furrows. And then next thing, viewing of this maize shown in that AM pit. So after that vermiculate contain that is of a raised AM infected the maize plants, you have seen that. Okay, so this is the main thing is carried out in that is of a in vivo. The worm culture is inoculated into the sparrows. Okay, the soil, maize plant cultivation. And then second one is called as in vitro. In vitro, it is called as axinic culture techniques. Axenic culture technique, there are three modes are there. What are they means? The solution culture, aerobonic culture, and root organ culture. So what is a solution culture? Solution culture means growing the infected root in the aqueous medium enriched with the mineral nutrient for a growth of roots under the controlled biotic and abiotic conditions. Abiotic means environmental factors, and biotic sunlight, all the features it come under the biotic condition. So these two conditions it promote the growth of a plant in the BAM. It is called as solution culture. And the second one is called as aerobonic culture. So aerobonic culture means you are taking the culture inoculum, and that inoculum you have to make the colonize for that is of a AM fungal inoculum production. So you have to apply on that aerobonic the culture formation. And then third one is root organ culture. Root organ culture means MS. MS rooting media is there, like a plant tissue culture. So MS rooting medium. This is the called modified agar media, and this liquid media for creation of increased amount of root from the callus tissue, and these roots are infected by AM spores or by the surface sterilized root which obtained from this mycorrhizae plant. Okay, so these are the tastes. So what are the crops which can be used for this? So the mycorrhizae species, different mycorrhizae species, especially used for the different crops. Barley, maize, and wheat, we have taken that of a glomus. And a bee means that is called as Asbulomycorrhizae, glomus, and Gingospora. And peanut, if you have taken the glomus and then fasciculatum and Synodonta decidi. Pea plant, if you have taken glomus, intratuberculous, and cotton, glomus species, and sclerocytosis sinuosa. And then tomato and potato varieties, the Gingospora, moramida, glomulus, and Aclospora. These are the different species of fungus. Okay, and black pepper, if it is called as Enteropasporia and Chlorominia and Scalodrium viviparia. And then cardamom, if you are taking glomus species, and then citrus also what? Glomus species, and then glomus mosaic, and marigold, sunflower like this marigold, and this marigold have that is of a glomus mosaic. So these are the different type of fungi. It is used in the plants, different variety of plant. The main thing is the plant protective mechanism, and another important as a point is defense and drought resistance, water intake is less. And then next thing is application of this BAM fungi. So where you are applied this? Carried out in the nursery applications, 100 gram of bulk of inoculum, it sufficient for 1 meter cube, 1 meter square, 1 meter square. This inoculum should be applied. What do you do that? First thing is below the soil, 2 to 3 centimeter you take that. Before sowing that everything you can make the bed. Okay, so 100 gram of bulk inoculum, you can pour it on that 2 to 3 centimeter below the soil before the sowing it. And the seeds or cutting should be sown and planted above the BAM inoculum to cause the infection. Whether the infection is carried out in the root or you check it. And then next case, commercial wise, you can take it in a smaller size polythene bag raised crops. So that cases 5 to 10 gram of bulk inoculum is sufficient for each packet. Mix the 10 kilogram of inoculum with 1,000 kilogram of sand potting mixture and pack the potting mixture in the polythene bag before sowing. So before sowing, you have to make it confirm that everything, the pack should be ready or not. The package should be ready. You can use it there. Okay. So this is the main important the feature of this. Okay. So the polythene bag, it is very useful, and then you have to use it for a large scale, especially for the seedling plant or the transplanting plant. You have to use this polythene bag raised crops. And then another important thing is outplanting also we have to use that. Outplanting means seedling case. So that seedling case you have concentration only vary. So 20 gram of BAM inoculum is required per seedling and apply this inoculum at the time of planting. And finally, for existing trees also you have to use the 200 gram of VAM inoculum is recovered for inoculating the one tree. So apply this inoculum near the root surface at the time of the fertilizer application. So fertilizer application time, especially at the root surface, you have to apply this inoculum. Okay, so these are the main important features of this application of this VAM. So what is it, VAM? VAM, it is the fungi, and the fungi is a symbiotic relationship with the root of a plant. And then the important as a feature is it has support to the growth of a plant. How it can be support the growth of a plant? The main thing is it have the phosphorus content. Okay, so it has supplied the phosphorus content. It gets the nutrient from the roots. It is a living inoculum, and this inside the root of a plant, and then through the root it occurs. And the next point is it have three important unique features there. So what are the unique feature means? The first one is the plant defense. Defense means it is against the pathogen. It is act as a insecticide, and then because the insects cannot come and enter the pathogen cannot come and enter into the plant. So it kills the pathogen. It acts as a barrier or it against, because it produces antibiotics. Antibiotic like substance produced automatically it suppress the growth of the pathogens. Therefore, it is a plant defense mechanism, especially of the coast defense mechanism is carried out. So that is the first point. And the second important thing is it support the growth of a plant, and then height, certain height also you can apply it. So the growth is also very high, crop quality and quantity, yield is also more, and best suitable for the small grains. Okay, and the next thing is the tolerance. So the tolerance, if you are talking about the tolerance, the two ways are the soil tolerance and that is of water. So the soil means they have the acidity feature. So the acidity feature also it is tolerated. The pH, if it has a pH is more, the plant can survive. If you are applying that as of a VAM. Okay, so acidity of a soil is tolerated, and then next thing is water. So in the stomata, the water transpiration mechanism is it is what happened. It is so less intake of water. So especially no need of water in certain plants also you have to apply this VAM. So finally I come to concluded that is of a VAM. It is called as a mycorrhizae. It is a live in the rhizosphere of the root. It support the growth of a plant and enhance the growth of the varieties of the crops and enhance the supporting the nutrients. It supplies the nutrient as well as the support the growth of a plant. Okay, so this is the main important as a feature is present in that, and then bring about the activity. And what activity means? That is of a it acts as a biofertilizer. Biofertilizer, living biofertilizers, inoculum. Either you have to apply onto the seedling or to the transplant. You have to apply it. So that is the main important nutrient source for a growth of a plant. Okay, so all the plant varieties you have to apply on that, but it cannot increase the nitrogen, phosphorus and potassium content. Okay, it support the growth of the plant. Okay, thank you so much.